All right. So, how's it going, everybody? We're doing our interview for Tropical Marine Ecology, and the scientist that we specifically chose to interview is Dr. Laura Gray. So, I know myself, I had you in bio back in 2020. I'm sure some of you guys also had that. And now so you're over at Hui now in the Marine and Geochemical unit. Is that correct?
Marine chemistry and geochemistry, yeah.
What it is, yeah. Do you want to
Yeah.
talk a little bit about that?
Yeah, so my current position, I am the digital coordinator for the Center for Chemical Currencies of a Microbial Planet, which is an NSF, National Science Foundation Science and Technology Center. And so essentially what that is that this center is based at Woods Hole Oceanographic Institution, but it actually involves scientists from, I think we're up to 14 or 15 different institutions around the country. Um, Also, one of our scientists is working actually in Bermuda at BIOS, Bermuda Institute of Ocean Sciences. And our research center is broadly focused on understanding how microbes cycle metabolites in the surface ocean.
Yeah, I'm sure you know this past summer, surface waters off of the southern tip of Florida, over by the Keys, reached record high sustained temperatures at about like 100 degrees in July from like the 25th to like the 27th. Can you speak on what this means for corals left in the region, if there are any left?
Yeah, so I guess I'll start with the last part of the question first. So there still are corals left in the Florida Keys. Um, but essentially when you hear that sea surface temperatures have gone up incredibly high and remain very high for a sustained period of time, that signals warning bells for coral bleaching, which is a process that you might have all learned about already. And I can just briefly recap it here. I won't go too much into detail because that's actually not my specific area of research. But coral bleaching essentially occurs when there's a breakdown of the relationship between the coral organism itself and the symbiodinium that it hosts. So symbiodinium are the photosynthetic algae that live inside the tropical coral organisms that I primarily focus my research on. And so there's something that I would say, I mean, there's a lot of people who study coral bleaching, but figuring out the causative effects of coral bleaching is actually quite incredibly difficult. And so I still don't think there's a widely accepted consensus on how bleaching occurs. But essentially what happens, like I said, is that there's something about elevated sea surface temperature that impacts the relationship between the coral and its symbiotic algae, and the coral rejects the algae. So we say bleaching occurs because the algae is what gives coral its pigmentation, its color. And so if the coral is rejecting the algae, and then all that you see is just the calcium carbonate skeleton underneath with a clear tissue over the surface of the coral. And so corals can survive. A lot of corals die, but some corals can survive if they're able to recover the microalgae over a period of time. But generally, we don't like to see that sea surface temperatures get high, especially that high and for that long. And so that's a huge stressor for coral reef ecosystems, is when they experience these high sea temperatures.
Mhm. Cool. And then my final question for you. So this is kind of based off of a field based survey you conducted in 2019, where you compared biomass, diversity, composition, and functional capacity of microorganisms surrounding scleractinian corals in the Cuban reef system. Can you explain kind of in simple terms how microbes interact with corals and why studying them is important for coral health?
Yeah, well, that's a really good question. I don't know if I can explain it simply because it's actually, like everything
Yeah.
in science, it's very complex, but I'll try. Um, so I would start by saying that, like in humans and other familiar organisms, microbes are essential for health, right? So in humans, we have a very diverse microbiome that resides within our gut and helps us digest our food. And every day we're learning more and more and more about how these microorganisms in in our bodies really impact health and functioning of our entire body, not just our digestion, but other parts of how our body functions. And so that just kind of underscores how important microbes are for the functioning of their host organisms. And so you can think about the same thing happening in corals, even though they seem to be a lot more simple than humans are or other animals. They are still animals, and they're completely covered in microbes from within and on their surfaces. So in coral cells, microbes live. They live in the lining of the gastrodermis of basically the coral stomach, and they live on the surface of the coral in the coral mucus layer. And in all of those environments, we think they play really important roles in the health and the growth of corals. So every day there's new research showing that a particular type of microbe makes, this vitamin that's important for coral growth or health. And so research has shown that microbes are important for nutrient cycling in corals, the symbiodinium themselves are important sources of sugars and carbohydrates for the coral organism. The coral actually uses for its own metabolism. There's some coral-associated microbes that create basically antibiotics that help protect the coral against infection from another pathogen, another microbe that one might want to opportunistically infect the coral itself, right? So more and more we're finding that these microbes, while their relationship with the coral is very complex, we think a lot of them are actually quite beneficial for a coral, and the coral, in fact, probably needs some of these microbes to grow and survive. But that's a huge field of coral reef microbial ecology is kind of a domain in and of itself. And then there are researchers who study just what goes on within a coral organism itself, within one organism or, a couple of different species of corals. And there's actually really interesting research now being done to try to figure out how to manipulate the coral microbiome to potentially see if they can make corals more tolerant to stress. For example, coral bleaching or, ocean acidification or higher nutrient concentrations. And so it's a really cool area of research that's occurring. And it's just trying to understand, all the ways that corals microbes are important for how corals live and grow.
Awesome. Thank you so much. I think now we're going to move on to Grace with her questions.
Hi. Um, so my question is, how does the knowledge that you just described about like the microbes and sponges, how can that be applied to conservation efforts?
Yeah, so a lot of my research is very exploratory and involved applying techniques that were, some of them have not been used at a grand scale in this particular environment. And so I would say that it's really hard to jump immediately from, a finding in one of my papers and then a general conservation, I guess, recommendation or requirement. But I think my contribution to the field is that I've really kind of helped add to the complexity of all the things that we need to understand about how coral reefs function, particularly at the intersection of corals and seawater microbes, right? So we know coral reef environments are incredibly complex. We have the microbes who are doing all sorts of things in the seawater overlying the coral reef, and then you have the coral reef itself that's populated with corals and sponges and algae and other organisms. You have fish that are living and pooping and feeding all over the coral reef. And all of those organisms, microbe and, macro, big organisms, macrofauna, contribute to energy cycling and nutrient cycling on the reef. And all of that feeds into the health of the reef and the balance of the nutrients that are available, the carbon that's cycled, all of that. So coral reefs are incredibly complicated ecosystems, but they're incredibly important for how for the ocean, for the health of the ocean, because they sustain there's some estimates they sustain up to 25% of all marine fish species. You have a tremendous amount of biodiversity that resides on the coral reef at one point or another during their lifetimes. And so I think my findings, they don't have a blanket recommendation for conservation efforts, but what I was really trying to do is to show, we can see these trends or patterns in how microbes interact with coral organisms themselves. And we know that if there's species-specific differences in how corals influence the microbes that live around them, then as coral reefs change, as they continue to degrade, as the coral species on a reef change, it's likely that we'll start seeing differences, and we have already seen differences in the microbes that you can detect on reefs. And this probably also means that those microbes are playing out different functional roles and ultimately likely changing how nutrients and energy are getting cycled on the reef itself. And so it's hard to study a system that's changing in front of your eyes. And that's something that I think a lot of scientists, even in other fields, are struggling with, is that, as humans, we've been changing the earth so rapidly. It's hard to know, it's hard to have like a start and an end point, because you're always measuring things against time and things are always changing. But I really think that you have to make a start. You have to start somewhere, right? And yeah, I think this research eventually, hopefully, will feed into larger scale conservation efforts. But at this point, I would say we know too little about really how to implement that at a wide scale. So at this point in conservation, there's a lot of research focused on trying to just get more coral colonies to grow on reefs. For example, in the Florida Keys, they have a lot of outplanting efforts where they basically try to grow corals in like a lab setting, and then they take those young corals and plant them back on the reef with the hope that that coral will survive and continue to survive and reproduce and help reseed the population of corals on those reefs. But I guess we're constantly trying to move in that direction of being able to figure out the better conservation efforts that allow us to do anything that we can to help save these reefs. So
Thank you. I think that was a great answer. And I loved how you said that this is the starting point, really. And that's why I want to thank you so much for your research, too, and your contribution to the field, because, we do need to conserve these very crucial ecological systems. So my next question is, in your 2022 study regarding the coral ecosphere, you investigated the discharge of metabolites by benthic organisms in the Caribbean, such as stony corals, octocorals, and an invasive encrusting red alga. Could you explain the significance of the organic chemical compounds released by the corals and how they may vary by species?
Yeah, so this was a really fun study to take part in. Because it directly emerged from the earlier work where I studied the 2019 paper, actually, where I was interested in trying to understand, what are these interactions that occur between corals and the surrounding seawater microbes. And so one of my ideas coming out of that study was that, potentially it's the metabolites. And these are small organic molecules that are released by all forms of life. We really think about metabolites like they're the currency of microbes. And so metabolites are organic molecules, but microbes use these organic molecules to communicate with each other, through quorum sensing or other types of interactions. Microbes are directly dependent on having specific metabolites for their own growth and nutrition. So microbes are constantly taking up metabolites and releasing metabolites. And so it's just this really dynamic perspective on microbial ecology, is to understand how the presence and absence and the composition of metabolites impacts the microbes that can live in a particular environment and how they interact with each other. And so I was interested in taking what I knew about metabolites and applying it to coral reefs, and particularly trying to understand what are the metabolites that corals and their associated microbes put out into the water, and then trying to understand how the seawater microbes that live in that environment, how they might be using those metabolites. and how those metabolites might impact the species composition of the microbes. And so this study involved going to the Virgin Islands in St. John, and we collected colonies of like 6 different species, some hard corals, there's scleractinian corals, some of the soft corals, octocorals, and then the invasive Ramicresta textilis encrusting algae that recently has been really increasing in abundance on reefs, particularly in the Virgin Islands. So we collected the actual colonies from the reef, and then we did seawater incubations on board a ship to basically collect the metabolites, the chemicals that those coral colonies and their microbes releasing. And then we were able to study what those metabolites were using mass spectrometry to both identify and to try to quantify the concentrations of some of those metabolites. And so essentially we found that corals and other benthic organisms are sources of these metabolites to the surrounding reef. And that's really important because reefs generally exist in very low nutrient seawater, and we would call it oligotrophic conditions. Although you have, like with everything in biology, there are exceptions to that rule, of course. But as a rule of thumb, coral reefs are typically found in these low nutrient tropical areas. So any source we think of new, we would call it labile or easily used by microbes, any source of labile metabolites is really important for the organisms and the microbes that live on the reef. And so through this study, we basically confirmed that yes, corals release some of these labile metabolites to the surrounding reef seawater. And so we did find differences by species, which suggests that not only do we know that corals and other organisms are releasing metabolites, but they're releasing different types of metabolites. And those different types of metabolites impact the microbes that respond to the metabolites and how those population dynamics change as well. And so we were trying to not only do a pretty comprehensive study, at least. We had 6 different species. We had really high replicates. So we had confidence that we were measuring what we thought we were measuring, but then we tried to put it into a more ecological context by measuring the response of some of these natural seawater microbes to these metabolites. And so we did find differences. And then, but we were only able to do kind of incubation-based studies. You can imagine that it's hard to take those results and apply them to a reef setting, but at least we were able to show that, yes, it does matter. Species composition does matter, that it impacts the metabolites that are released and how microbes respond to them and how microbial populations change as well. And so it was a really fun study to do. And I think that area in and of itself is a big area of research that I would hope that more people will go into as time goes on. Understanding how these metabolites impact coral reef microbes and how they interact with each other.
Thank you so much. You actually answered my follow-up question as well.
Oh.
So I'm going to pass the baton to Roy.
Okay.
Hello, my name's Thomas
Hi.
Roy. I also had you for biology freshman year as well. So my first question was that in 2019, you took part in the comprehensive study examining the microbial community of Northern Caribbean reefs, variant in human impact and protection in Cuba and Florida Keys. Could you speak on the differences observed between heavily protected offshore reefs in Cuba and the more impacted nearshore reefs within the Keys, and what specific factors contribute to the disparities?
Yeah, so this is a really good question, and it's really important for context for that study. So we originally collected samples and went on cruises in 2015. And so that, now that I think about it, that was a while ago. But Jardines de la Reina is the heavily protected reef system that's south of the island of Cuba. I think it's about 60 miles south of the mainland of Cuba. And so a couple things to note, just by geography, the reef systems are pretty far away from other land masses, and it's harder to get to, for one, because it's 60 miles away from the mainland of Cuba. You also have between the mainland of Cuba and the reef system itself, there are really healthy mangrove environments and seagrass environments that help basically, well, I think there's a couple studies that have shown that the buffering of those environments basically help protect the reef system itself from any land development or like runoff that could be coming off from the mainland of Cuba. So in addition to that being very far away, you have all these other environments that help kind of minimize disturbance of the reefs that are in the reef system in the archipelago. The other thing to note, at least at the time, was that Cuba doesn't really have large scale industrialized agriculture. And so that also feeds into, not having as much nutrient pollution, perhaps, that could get out to the reefs. And the other really unique thing about this reef system is that there's no takes. It's like a no-take zone. So, for example, you can't go and fish there, and that's actually enforced. So there are a lot of places that across the world where you're not allowed to take fish, but the enforcement is really difficult because you have to have people to be out on the water to watch, to make sure, to patrol, make sure there's no illegal fishing going on. However, because they're less stressed from direct human impact, that means that potentially they're more resilient to dealing with some of these other stressors. And so it was really cool to dive on those reefs, because I've never been on reefs like that, that are, so healthy compared to reefs in the Florida Keys, for example, and that have lots of fish on them. And so that to be able to just see that in person was really amazing. So those reefs are quite different than the reefs in the Florida Keys. And you can imagine why the reefs in the Florida Keys have been more historically impacted by humans. They're part of the Florida Keys National Marine Sanctuary, but that wasn't instituted. I think it was started in the 90s to try to protect the Florida Keys. But by that point, they had been, exposed to a lot of direct human influence. There's a lot of land development that occurs near the coast in the Florida Keys. The reefs in the Florida Keys are pretty close to the coastline. And people are able to access the reefs a lot more readily than the reefs in Jardines de la Reina. Oh, the other thing that I should have mentioned is that I'm not sure what the number is now, but they limit the number of people who can visit the reef system, the reefs in Jardines de la Reina. And I don't think they do that for the Florida Keys either. So generally the Florida Keys are just more impacted by humans, and they have been historically for a much longer amount of time. The reefs are closer to land. There's more pollution that causes eutrophication that occurs. And there are particular zones of the Florida Keys National Marine Sanctuary where no fishing is allowed, of course, and that helps. But these reef systems have been historically more impacted than Jardines de la Reina. So I think that's a big difference there in terms of level of protection. The other thing I will mention is that reefs in the Florida Keys have fairly recently been plagued by stony tissue coral loss disease, which is a major coral disease that I think started spreading through out of Biscayne Bay through the rest of the Florida Keys. And I want to say 2019 is when they first started noticing it. And it's a pretty serious disease. It impacts a lot of different coral species, and it can kill big colonies in a matter of weeks. And so in 2019, or maybe it was 2018 when it started, because I did research in 2019 in the Florida Keys. When we went on a field excursion there, we saw, these huge coral colonies that were just dead because this disease had come through and wiped them out in a matter of weeks. And that's really hard to see. But disease is a big threat to coral reefs as well. Disease is also more typically present on reefs that are historically and more likely plagued by direct and indirect human influences as well. So.
Well, thank you very much. That was really interesting. And so for my last question, so the region of study for this project was the Keys. Looking at 5 different coral species, do you think it would be useful to do a similar study, looking at tissue, mucus, holobiont
Mhm.
habitants in different regions of the ocean, such as the Great Barrier Reef or the Indo-Pacific?
Yeah, I mean, I think I'm an advocate for as many studies as possible so we can get a sense of some of these more global trends in terms of um, the microbes that live in association with corals. Are there specific microbes that are more commonly found in corals versus others? Does that occur globally, or is that more of a regional thing?
Great, thank you very much.
Sure.
I'll turn it over to Zach now.
Hi, how are you?
Good. How are you doing?
Good. So for my first question, I was going to look into the study you did of distinguishing between microbial habitats and then the ecological complexity in the coral biomes. When this was published in 2016, you mentioned the topic of metabolic interactions between the archaea, bacteria, and coral, and how it was not that well understood. Now looking like 7 years later in 2023, do you think those interactions are better understood at all, or is there still like a lot of research to be done on that?
Yeah, that's a good question. I think we have a better handle on it, and I think over time we've been able to develop the techniques and really get more of a sense of what are we collecting, what are we measuring, how can we measure it at a smaller scale. But I would say that I think there's a lot more to do, and I think in science, there's no limit actually to asking questions. I think you have to impose a limit on yourself just so you don't go down a whole bunch of different directions, just for your sheer sanity. But I think once you do a study, then you ask more questions about it, or like a result doesn't look the way you thought it would, and you want to understand why that is, and then you would do another study to try to address that. And so I think you could just keep asking questions, and I think that we still need to just keep asking questions and trying to make these observations along the way. I mean, I do think that over the course of my PhD, the field has changed in terms of the knowledge that we have gained. Also, the severity of the impact of climate change on coral reef ecosystems has grown. I think compared to, we've known it was a problem, like when I started at Hui in 2013. But now, every couple of days you hear about some other huge bleaching event or a disease that's killing thousands of different coral colonies. So I think there is need for a lot more research, especially in the applied research. Getting back to Gracie's question about conservation efforts, at some point we need to be able to merge the exploratory research with more focused conservation efforts that directly are applied. And that's really difficult to do, because sometimes science moves at a slow pace. And it's hard to keep up with all the changes that we're experiencing, especially on coral reefs. But I would say there's always need for more information. And I think that's one of the exciting things about science. So
Yeah, thank you. And then going on to my next question, the final question is, what advice would you give to any student at Mass Maritime who are interested in pursuing a career in research?
Yeah, that's a good question. So I would say the first thing is take a research project from start to finish and see how you like that. Because it's one thing to design an experiment, it's another to run the experiment, and it's completely another to analyze the results, use statistics to try to understand what you're seeing, and then write all that information up and communicate that to other scientists or to a larger audience. And that will be a really good indicator of whether or not research is what you would like to pursue.
Sweet, thank you so much.
Sure.
that's all the questions I think we have. Thank you so much for your time. I appreciate it.
Sure. Yeah. Any other questions or?
I think we're all set, but I really just want to drive home how grateful we are for your participation in our
Aw,
project.
sure.